Hello and good afternoon friends, welcome once again to the CEC-UGC live lecture. Dear friends, in this session today we are going to discuss about bio aerosols. We would be understanding what basically bio aerosols are, and for this we have again with us in our studios Dr. Arun Srivastav. He is an assistant professor in School of Environmental Sciences, Jawaharlal Nehru University. So first of all I would like to welcome our guest Dr. Srivastav, and I hope that with the help of his experience we would be able to learn more about bio aerosols. So welcome to the EduSet lecture, sir. We have heard a lot about aerosols, but what are bio aerosols?
Well, thank you, and today in fact the topic is bio aerosols and we will be discussing on this bio aerosols. Good afternoon everyone. So as the name indicates, it's the combination of two words, bio plus aerosols. So far we had been hearing and we had been discussing about the aerosols, mainly physical and chemical characteristics of aerosols. So bioaerosols are basically a different class of pollutants which comprises of different biological agents, and its effect is definitely biogenic. So starting from its definitions, what are bioaerosols? Bioaerosols basically are short form of biological aerosols, and bioaerosols are airborne particles that are biological in nature mainly, and these can be living or dead. And either if it is not living, I mean if they are dead, so they are generally originated either from living or dead organisms. Bioaerosols react to the air currents and move quickly or slowly depending upon the environment, and these particles are of very small size range, and generally their size is less than, it starts from 1 micrometer, that is 0.0004 inch to 100 microns, they are 0.004 inches. So the smallest of the bioaerosol among the bioaerosols could be viruses, which are of the size of 10 nanometers, while the largest could be pollen grains, which are of the size of 100 micrometers. And the atmospheric lifetime of bioaerosols can range from near indefinite time for some viruses to few hours for the larger pollen grains. I mean some of the bioaerosols, especially those which are of pollen originated from some plants like pollens etc., their lifespan or their stay time in the atmosphere could be for a few hours, while the bioaerosols which comprised of viruses and their lifespan could be for it could be of indefinite time, maybe for some days or some weeks or for some months too. And bioaerosols are broadly classified in two types. They are viable and non-viable bioaerosols. Now I will discuss one by one what are viable aerosols and what are non-viable aerosols. Viable aerosols are basically living organisms. I mean those aerosols which we discussed earlier or in the definition of bioaerosols, that bioaerosols are of two types, it could be living or dead. Similarly here the viable aerosols are those aerosols which are living organisms and they in fact demonstrate the microbiological activities, and they have the potential to multiply, or they can grow, they can increase their population from few to hundreds or thousands in number, and these includes airborne bacteria, fungi or viruses. Among these bacteria and fungal spores are the two most prevalent bioaerosols present. I mean in these viable bioaerosols the most prevalent are fungal spores and bacteria. Viable bioaerosols can cause acute as well as chronic diseases. They are very harmful in fact. Therefore, viable bioaerosols may be termed as infectious or pathogenic, whilst the non-viable aerosols are not currently alive. I mean they are generally dead. Therefore, they cannot multiply, and the examples of these bioaerosols are aerosolized pollen, animal dander, saliva and insects excreta. Non-viable bioaerosols are generally do not cause diseases, but can cause allergic and start toxic reactions in body. Non-viable bioaerosols are generally termed as allergic or toxic bioaerosol. In fact, these are the bioaerosols, they do not directly cause any harm to the body, but their chemistry, in fact their composition or the secretion they do, they are harmful to the human body. In fact, those secreted chemicals, they are pathogenic or they are usually harmful or toxic to the human body. Now, as far as the sizes of bioaerosols are concerned, I earlier told that the smallest could be few nanometers to up to few microns. So, in fact, here we can see that the size of particles vary from 0.001 micrometer to up to 1000 micrometers, and they include viruses, bacteria, fungal spores and pollen. Here we can see the smallest are viruses, and their size in fact vary from 0.01 to approximately 1 micron, while the bacteria size, they are generally bigger than the virus, and their size vary from 0.5 micron to up to or slightly over 10 microns, while the fungal spore size vary from 1 micron to up to 100 microns, while the largest one, they are the pollen, and their size generally vary from 10 microns to up to 100 microns. Apart from these, their particles, their bioaerosols particles are the combination of multi-phase particles, which can even be of bigger size than that of 100 micron. Now, what are the examples of viable bioaerosols? The examples of viable bioaerosols, I earlier told that mainly they were virus, bacteria, but apart from that, virus, bacteria, fungi, protozoa and algae, all these come into the viable bioaerosol, means they have the implication to the health as far as the infection and toxicity etc. are concerned. They have the capacity to grow further and further, they have the capacity to multiply, they have the capacity to increase their population. And as far as the example of non-viable bioaerosols are concerned, they are pollen, animal dander, saliva, insect excreta, endotoxins and mycotoxins. The later two examples like endotoxins and mycotoxins, they are very toxic in nature and they are really harmful to the human body. They are not, they are in fact not alive bioaerosols, but they are either secreted or they are found in the cells of the bioaerosols. Now, how this, so far we discussed about what are bioaerosols, what are the types of bioaerosols, what they can do, and now we will discuss how these bioaerosols can be collected to for further investigations. As far as their collection is concerned, they can be collected by two different sampling methods. One is inertial sampling technique, another is non-inertial sampling technique. Now, what is inertial sampling technique? Inertial sampling technique, in this type of sampling, we apply the inertial property of bioaerosols for their collection. I mean, inertial property, we all know that the movement or if the particle is moving or if the particle is stationary, it wants or it continues to move or it continues to be stationary, but when we hamper that characteristic to collect them, and when the particles are collected by virtue of that property, it is known as inertial sampling. In this type of sampling, acceleration or deacceleration play a very important role as far as its collection is concerned. Thus, the way in which particles are collected is highly dependent upon their size. I mean, in case of inertial sampling, we can be size selective as far as the particles are concerned. This is why this is also known as active sampling. While in case of non-inertial sampling, the sampling method do not rely upon the property of inertia of the airborne particles. There is no use of acceleration. Thus, the way in which particles are collected is less dependent on their size. So, here we can never be size selective, or the particles which are collected, it is not having any size selection criteria. This is why this is also known as passive sampling. What are the disadvantages of non-inertial sampling? Since non-inertial sampling is a kind of passive sampling, this is why it is non-volumetric. It means that the amount of air which is being passed through or which is supposed to be passed through to have information about the volume of the air, we cannot have information about that. Or finally, if we want to represent the concentration of Bio aerosols in the terms of per cubic meter volume of air or per liter volume of air, it is not possible in these type of cases. And another disadvantage of these type of samplings are that it over represents larger particles sampled from a larger volume of air during the exposure period because of fastest sedimentation rate. We do not have any control on the volume of air in such type of collection techniques. Now, what are different types of inertial sampling or inertial samplers? They are impactors, and another one is centrifugal sampler. Now, starting from impactors, what happens in impactors? In fact, the collection by impaction relies on the tendency of particles to deviate from the air stream because of inertia. When the airflow streamlines bend to bypass a solid or semi-solid surface. Here in this picture, we will discuss elaborate about these impactor techniques with schematic diagrams. First, what are the different type of impactors? They are sieve impactors, or they are also called as sieve samplers, cascade impactors, slit samplers, and impingers. Now, we will discuss one by one. What are sieve samplers actually? Sieve samplers are devices which use the acceleration of air through numerous small holes of definite diameter. They are also called as inlet orifices in a perforated plate, which in turn impacts suspended particles in the air onto the surface of the agar. They can be of two types, one is single stage and another is multi-stage, or this is also called staked sieves. One stage samplers have only one perforated plate set in front of one agar plate, while staked sieve samplers can have up to 6 stages of perforated plates and agar plates. Now, this will be clear with this picture. In this top left one represents the sieve sampler. In fact, this is one stage sieve sampler, and here we will see in the middle one there is a petri dish having a medium. This medium is basically agar medium, where the particles actually are collected, and in the bottom picture, there is only one picture, we can see with the schematic that air is passing from the top and it in fact first collides with the agar plates, and what happens during the collision, the particles are deposited onto that surface, and from there the air stream is reflected, and with a narrow stream it is going outside. So, we can see here there is a cascade effect in the sampler, and with that cascade effect the particles are deposited onto the agar plate, and the right side there is a complete picture of the sampler. This in fact is equipped with a motor or pump tube. So, this was the example of one stage sieve sampler. Now, what happens in multiple stage sieve samplers? We can see here, this is in fact accumulation of many one stage sieve samplers. Here in this particular picture, we have a 6 stage sieve sampler, or we can see there are different 6 petri plates and there are different 6 perforated dishes. So, now we can see from top to bottom what happens. Again it is being followed the same procedure what was done in the previous one, but here it is basically a combination of multiple stage. So, that effect is repeated continuously 6 times. So, what happens in the first stage, when the air stream comes and it strikes on the agar plate, and again the same cascade effects take place and the particles are deposited onto that agar plate, and later on it passes through the second stage, and there again that air collides with the second stage or the petri plates on the second stage, and there it further deposits some particles, and from there the again cascade effect is done and the air stream passes through those narrow stream and it goes to the next stage. So, what happens, there is a continuous impaction as well as continuous deposition on the particles on different, or we can say or on all the stages, and thus the advantage of this type of system, one beautiful advantage of this type of systems is that we can get particles in segregated manner, or in other words we can say that the particles deposition are not uniform, rather they are deposited in decreasing size manner. So, in the stage 1 the particles deposited will be of the coarser size range, or it will be the biggest as far as their diameter is concerned, and their diameter keeps on continuously decreasing further and further. So, the particles at the last stage will be of the smallest diameter. Thus, we will have in this fashion we can get particles of different 6 size ranges. Now, what happens here is the explanation of the same thing. We can say that air enters through the top of the sampler and larger particles are impacted upon the surface of the petri dish of stage 1, and later on the smaller particles which lacks sufficient impaction potential follow the air stream to the subsequent level. As the air stream passes through each stage, air velocity increases, thus increasing the impaction potential, so that particles are trapped on each level based upon their size. Therefore, larger particles are trapped efficiently on stage 1 and slightly smaller particles on stage 2, and so on until smallest particles are trapped on stage 6. In this way, the particles are segregated based on their size. Now, what are slit samplers? In fact, in a slit sampler, air is sucked through a narrow slit that is 0.25 millimeter wide and 27.5 millimeter long and 3 millimeter deep onto the surface of agar or other culture medium or other required culture medium contained in a petri dish placed just below the slit. Here the picture of that same that type of sampling technique. Here the top left one, we can see top left, and in fact all top left, right, and the bottom right. It is a complete picture of the slit sampler, but here we can see in the first left picture there is a combination of two things. One is a plate and dish, we can say dish having a slit, narrow slit, and just below that we have agar plate or the medium where the particles are supposed to be collected, and the exact picture or the exact picture of the sampler is given on the right top picture. Here this top dish is in fact combination of or having many slits radially to that circular dish, and in the top bottom right bottom we have the picture with the motor 2, and the bottom left picture it actually shows the deposited particles in a very fashioned manner. Here the particles are deposited readily onto the agar plate. Now, the next type of samplers are impinger type of samplers. In impingers, air is drawn by vacuum through a body of liquid and particles leaves the air stream to be collected by impingement into the liquid. Liquid can be water, broth, mineral or oil. Broth or mineral oil. Most impingers are made from glass with single collection chamber and suction applied to a side of. impingers can be of two types, one is single stage or the another is multi-stage or fractionated size impingers. Now, in fact, here how the impingers can be classified, they are of two types, single stage and multi-stage. We can see here the pictures of single stage impingers. Here the extreme left picture actually shows the outlined picture of outline diagram of what happens actually. The red color arrowed line, curved line basically shows the flow of incoming flow of air, and this in fact this impinger or the tube contains the required liquid. What it could be either water, mineral oil or broth. So, the tube which contains the liquid, that particular or required liquid, there actually the air which is of red color, represented in red color here, strikes upon it, and what happens after striking, the air returns back, which is shown here in blue color, and it passes out. During this impaction and returning back, what does it do? The particles actually gets deposited onto that liquid, and this further can be analyzed for the estimation or for the identification of Bio aerosol. Here actually given the schematic, the complete schematic of the impinger tubes, and the picture in the middle one, it shows the what are the tubings etc., and the picture at the extreme right is basically impinger combined with a suction pump, because suction pump is also a mandatory part for any Bio aerosol sampling technique. Now, what is multi-stage impinger tubes? Here there are different three types of in fact different two types of multi-stage impactors are shown. One is 3-stage impactor, while the another is of 5-stage impactor. So, what happens if we see the picture at the extreme right, we will see the air passes through the top opening and it goes into the liquid, and there it deposits some particles, and afterwards after colliding or after reacting with that liquid, the remaining air or the air comes out, and it then passes through another hole or another opening and it goes to the stage 2, and there again it gives or deposit some particle, and there after depositing the air comes to the third stage, and so on it keeps on doing. So, in this fashion, because there is again energy loss of the particles and the increase in velocity, so the particles are again segregated in a very particles are again segregated with size ranges, with different size ranges, and finally air passes through the outlet which is at the bottom of the right side picture. So, in this way the particles are segregated in different 5 sizes. The next class of sampler is centrifugal sampler, and in fact centrifugal samplers are divided into 2 different type of samplers. One is known as cyclone samplers, while another is called as centrifugal sampler. Now, here what happens in case of cyclone sampler, air enters through, in fact the picture at the left hand side beautifully shows this. The red color arrow or curved line basically shows how the air enters, and the blue color shows how the air passes through it. So, what happens, or the remaining 4 pictures actually shows there is a tubing mechanism having some liquid or the required medium, it could be liquid, broth again, or the mineral oil, and this is represented here in light blue color, very light blue color. So, now you can see how air is entering inside it, and after depositing the particles, it is coming out through the opening at the top, and the particles here deposited are shown in the orange color dots, and there is, and finally we can say in the picture 4 at the right hand side, the particles are deposited, they are represented by orange dots, and in fact the particles deposited are also encircled in orange color. So, now we can, as far as definition is concerned, we can say that in cyclone samplers a vortex is created. Vortex is basically whirling of the particles so that particles can efficiently be deposited into the liquid. So, a vortex is created in which particles with sufficient inertia leaves the air stream to be impacted upon a collection surface, and from this mechanism this liquid or the fluid which contains the bioaerosol or the aerosols particle containing aerosols particle of biogenic nature are actually accumulated in that liquid, and that liquid is taken out for further investigation or further information about the bioaerosols. Now, here is basically picture of some of the bioaerosol samplers, and in fact these bioaerosol samplers are of usually low volume sampler. They are not for heavy uses and they are best suited as far as the personal exposure is concerned. So, these samplers can efficiently or very nicely be installed onto the collars of some person equipped with a motor or pump, and there over the time we can have some information about or some concrete information about, or we can correlate it with the inhalation of that particular person who has installed it on his or her collar. Now, what are the centrifugal samplers? In fact, in centrifugal samplers, air is drawn on an impeller housed inside an open shallow drum, afterwards accelerated by centrifugal force towards the inner wall of the drum. Inner wall is a plastic strip supporting a thin layer of agar medium onto which airborne particles are impacted. These strips are then removed and incubated to reveal microbial colonies for deposition. Now, here the picture is shown. Here what happens is the top picture will see the schematic or the outlined diagram of such things. Like what happens, particles are basically there is a whirling kind of situation inside the sampler, and during that whirling the particles or the air stream basically having particles or containing particles collide with the wall of that particular sampler. In fact, the walls of these type of samplers should be layered with some specific medium. It could be agar or any other required medium. So, the particles or the air stream, when it strikes onto that medium which is along the walls of the samplers, these particles get or they are deposited onto that wall or that medium actually, and afterwards that medium is taken out and it can be utilized for further investigation. At the bottom two pictures is a complete picture of centrifugal samplers. Now, the next one is non-inertial type of sampler. As far as non-inertial type of samplers are concerned, they are again of many types. They could be gravitational, or it is also called as settle media, biotapes, filtration, electrostatic precipitators, thermal precipitators, and condensation traps. Now, here what is gravitational or settle plate? This is basically, in fact, this is the simplest form of collection of biological particles. In fact, in this type of samplers or techniques, an adhesive substrate such as microscope slide or petri dish containing agar medium exposed to the atmosphere to collect particles by gravity. This method does not require any specialist equipment. So, what is done in this type of techniques actually? A petri plates containing agar media is exposed into the atmosphere or it is kept somewhere for some times. So, what happens? The air or the particles with the help of gravity which falls upon it, what does it do? It deposits some other particles over here over that particular surface, and later on that surface is utilized for the investigation or for further growth, etc. So, in these type of techniques we cannot have much information about the volumetric concentration. And in fact, in the bottom right side pictures there is a growth of colonies, and it is we can see there is an increase of colonies from 100 to around 1 million in due course of time. Now, the next class of such type of examples are biotubes. So, we can say that biotubes provide a standardized surface sampling method used for determination of possible microbial bioaerosol contamination. It provides the ability to quickly take a sample and measure the relative degree of contamination. It is especially useful when sampling valuable or non-transferable materials and is very effective on smooth areas of concern. In fact, these type of samplings can be where it is not possible to take the samplers, or in fact those places where the deposition have already taken place. I mean, in such type of cases we are not much concerned with the air containing bioaerosols, rather we are concerned with the surfaces where the bioaerosols have already been deposited. So, what do we do in such cases? We actually have a tape having some adhesive material plus some media which can be helpful for their growth. So, what is done, that tape is actually kept onto that surface and pressed for some times, and afterwards that is taken away with the bioaerosol into it, and later on that tape is further utilized for further growth and further investigations about the bioaerosols. Now, the next class of technique is filtration technique. Filtration is a very simple and very general technique. So, what is done in that type of technique? Fungal spores are readily captured on filters. So, it is a simple pumping and filtering mechanism. Air is passed through a filter, and whatever the particles present in the air, whether they are biogenic or non-biogenic, they are deposited onto the filter papers, and later on that filter paper is utilized for the assessment or estimation of that if we are concerned with the biological particles. So, that filter is later on used or utilized for the assessment and estimation of bioaerosols. And in fact, now as far as its definition etc. are concerned, we can say that fungal spores are readily captured on filters, and capture is caused almost entirely by interception except for filters with very large pore spaces. Filters with pore size in the range of 1.5 to 2 micrometer are very effective at capturing more than 99% of airborne fungal spores. Using the largest possible pore size will reduce resistance and allow the use of small personal pumps at reasonably high flow rates. In fact, filters are also of different three types. They are subdivided into fibrous filters, membrane filters, and flat filters. As far fibrous filters are concerned, these type of filters include paper, cellulose, and glass fiber. The fiber forms a random crisscross matrix through which particles laden air stream is directed. The pores of fiber filters have range of sizes of which means defines the pore sizes. Now, here in this particular picture we have the example of fiber filter. At the left hand side we have a general picture and it is not magnified, the general filter picture, and it is not magnified, while at the right hand side there is the magnified picture of fibrous filters. Here we can see that the filters or the threads of the cellulose are arranged in a very crisscross manner. So, it in fact having some sieve type of structure through which the air is passed, and in fact these sieves basically decides what diameter size particles are will be deposited. And the next class of filters are membrane filters. Membrane filters are formed from cellulose acetate or cellulose nitrate, PVC, PTFE, nylon or gelatin gels. The pores are created by evaporation of solvents. The air stream follows a twisted route and thus the particles get trapped in and on the surface of the filter. Here the pictures of beautiful pictures of membrane filters are provided. We can see at the left hand side is the picture of the filters which is broadly used and is not magnified, while the three pictures at the right hand side are magnified pictures of membrane filters, and they are of in fact 3 different types as far as their composition is concerned. They are polypropylene, cellulose acetate, nylon membrane. Now, we can see they are of different as far as their sieves and structure etc. are concerned or threads are concerned. In first there is thread, whilst the another 2 there are no threads. They are, it seems that they are been grown somewhere. So, these are utilized in different type of Bio Aerosol samplings or whatever the requirement is. The next class of filter is flat filters, and in fact flat filters are made of polymer or metal through which holes have been etched. Examples are silver aluminum oxide and polycarbonate. In these type of filters, particles are collected on the surface rather than being trapped within it. For this reason, flat filters are most common media for Bio Aerosol, especially microorganism samples. There are pictures of 3 different type of filter papers. They are in fact arranged in such a manner that air is not passed through it, but air goes striking upon it. Thus, the particles are getting as far as particles instead of getting interlocked into it, they get deposited upon it. So, it is very easy to remove them from their surface. There are examples of the 3 different pictures of such filters are provided. Now, the next type of sampling technique is electrostatic precipitator. It is a very important class of sampling technique. In this method, particles are separated from air stream by electric rather than inertial forces, because so far we discussed about inertia or non-inertia in this class of sampling. In this class of sampling, inertia instead of inertia or non-inertia, the electric character is applied for the separation of particles. Therefore, collection is achieved with little pressure drop and relatively small amount of power. So, in this type of cases, we do not require much power or a heavy vacuum pump. So, with just some charges are required. So, the requirement of power is significantly less in such type of conditions, and in fact a vacuum source draws air stream into the sampler housing a high voltage electric field or corona discharge, and as particles in the air stream pass through the corona, they acquire an electrical charge, causing them to be attracted to a positively charged metal surface. This method collects Bio Aerosol into liquid, or in the final stage this method actually collects the Bio Aerosol into liquid or filter paper. So, here the picture of electrostatic precipitators are given. At the left hand side, we will see the outlined picture of electrostatic precipitators sampler, while on the right hand side there are 2 pictures of electrostatic precipitator, in one the combined, and while the other one is another one shows the inside of the electrostatic precipitators. So, what happens here? We can see in this picture at the top there is an inlet where from where the air pass inside, and afterwards we will see the air is in the middle. There are 2 outlets through which the air goes out, but at the middle there is a circular structure, or that is also in fact is also we can see there is an electric field. So, there is an electric field, heavy voltage electric field is collected, that is also known as corona discharge. So, what happens during the passage of the air, this corona discharge actually segregates the particles according to its charge, whether it is positive or negative, and that the freshly charged particles are deposited on another type of charged particles, from where they are efficiently collected or removed. So, these were the type of electrostatic precipitator. The next one is thermal precipitator. In thermal precipitators, dust laden particles through a narrow channel in which there is a temperature gradient perpendicular to the direction of the air stream. Particles entering this temperature gradient move in the direction of decreasing temperature. I mean, it is a kind of thermophoretic action, and the hot plate is the top plate, which is at the higher temperature or at the highest temperature is at 125 degree centigrades, whilst the coldest or the coolest plate is at around room temperature. Here the schematic of such type of precipitator is given. We can see here that there is a schematic, and the air is passing from the left hand side, and while it is getting out through the right hand side. So, and the middle one, there are two different plates. One is at a very hot temperature or at a very high temperature, while the plate at the bottom one is at a very low, comparatively low temperature. So, what happens, and in fact, because of this, a temperature gradient is created, and what happens because of this, particles get deflected towards the cool surface or the cold surface, and they are funnel type of structure. From after getting deposited on the cool surface, the particles are taken out or taken away. So, this is how the particles might be collected. Now, the next class of samplers are condensation traps. In condensation traps, Bio Aerosols act as condensation nuclei around which condensation takes place. After achieving a certain weight or size, the grown particles settle at the and then collected. So, it is the same kind of condition like what is there in the clouds. So, what happens, there is a particle, let it be the particular Bio Aerosol of interest, and there condensation, artificial or induced condensations are imposed upon it, and the particle or the Bio Aerosol particle, which is actually which initially works as condensation nuclei around which the water vapor is deposited, and it keeps on growing and growing, and when it becomes of sufficient size or enough size, it gets deposited on the surface. Here the schematic of the condensation trap is given. We can see here there is inlet and there is outlet, and in between there is a condenser. So, what happens, the particles while passing through condenser, it grows sufficient enough so that it can easily be removed away from the air stream. I think so we now today end with the sampling techniques, and the next session we will resume with the assessment and its pathogenicity, etc. So far we discussed about the different methods of collecting the Bio Aerosol, how the Bio Aerosol can be collected and later on we will resume with how they can be analyzed or further investigated, because it is also very important to understand their estimation, their understanding, their characteristics as well as their pathogenicity, what type of diseases, what type of harmful effects they can impose upon human beings. So, with this I think now if you have some further clarifications.
Definitely, sir. As you explained us in detail what Bio Aerosols are, let's revise what we studied in today's lecture. So, we would like to ask you again, if we talk about Bio Aerosols in terms of a common man, what Bio Aerosols are?
In fact, I told aerosols are, I think I have already taken a class on aerosols. Aerosols so far we studied or we discussed as its chemical as well as its physical properties. I mean the chemistry it has. the different metals, different chemicals compound, whether organic or inorganic compound. So these inorganic or organic compound can be harmful to our body when they are inhaled, but we always forget that these particles, apart from the chemistry, they have some biology also. I mean, they might contain some either living organisms or some dead organisms. If they are living organisms, if they are inhaled into our body, they can grow further and further, or they can be multiplied inside our body, that is also called as, or they can come in the form of infectious diseases, and later on when they are inhaled by us, although they were from the air stream or they were airborne, but once if it is inside the human body or if it has caused the pathogenicity, then that particular human body becomes a source to other human beings. Thus this is basically the biology of the particles which are so much harmful or which are of much concern. So in fact, we can say in a very ordinary word, like if someone sneezes, so people say that one should be cautious enough to put a handkerchief because the air which is coming out of our body is comprised of many viruses or bacteria. So if those viruses or bacteria which are airborne, if they are inhaled by the persons who are around you, it can harm those people. Thus in a very simplistic manner, we can say the air which has some biogenic particles, whether they are living or non-living, are known as bioaerosols.
You have also explained the assessment method for the bioaerosols. Please explain us in a very few words, because we have very less time left. So what are these
assessment
methods?
In fact, so far we discussed the sampling methods, how the bioaerosols can be sampled or can be collected, because our first aim is to collect them, and later on after collecting them, it's very difficult, it's not easy to collect the bioaerosol particles because they are in atmosphere. They are of many nature, they are of various nature, they are of different sizes. So our primary aim should be that it should be collected efficiently and nicely. Once they are collected, the assessment methods of bioaerosols are applied, and in fact these assessment methods etc. we will discuss in tomorrow's class. So I think we'll discuss more.
Yes, sir, sure, definitely. We would be discussing more on the bioaerosols tomorrow, and I would like to tell you all that Dr. Arun Srivastava would be again with us tomorrow. So if you have not able to understand certain points regarding the bioaerosols, I hope that the elaborative form would be tomorrow again. And as well as I would like to tell you all that you can access this particular lecture with the help of the YouTube, as we daily upload our lectures on YouTube. Thank you, sir. Thank you so very much.